Okay, so what I thought I would do is tell you how we, in my laboratory over the years, have used FRET and other biophysical measurements to explore a problem that actually happens to be very close to my heart, which is explore the problem of understanding the structure of membranes of living cells. Okay, so before I start, let me just provide a glimpse of the people who have been involved in this work, especially more recently. That's Madan, who's a close colleague and friend and collaborator. He's a soft matter physicist who's really been extremely instrumental in providing key insights into how some of these systems work at I think crucial moments in our journey here. So, and I'm going to bring those points out. We've worked with a number of his students. There's Kripa, Anirban, and this name here should have been Sarasej, 3 of Madan's students, and others who have worked in the stories that I'm going to tell you are Darius, Kabir, Abrar, Ambika, Riya, Subhrajit. Then we go on the right, there's Parijat, Chandrima, Thomas, and I think that may be it. Yeah, for this set of work that I'm going to talk to you about, that's probably it at the moment. Maybe Joey, who is this last person here. I'm going to talk about his work, but in not great detail. Anyway, the reason we're interested in the cell is because it's just very simply exemplified by this movie here. The cell here, you have a neutrophil that is running around, going after a bacterium, following it as if it knows exactly where it is. And the reason it's able to do this is because the neutrophil itself is integrating information from the outside, calibrating where this beast is running about, and redirecting, if you will, its whole self towards moving to this object. And this is completely determined by what processing is happening at the cell membrane. And the cell membrane itself is, is something that is quite remarkable. It's here you see it as a fluorescently labeled surface of a cell, and that cell membrane that we all know is consisting of multiple, maybe 1,000 different lipid species, 1,000 proteins, and it's completely in a dynamic state, right? But yet, I would argue that it's the cell membrane that is able to sense and respond, make the cell sense and respond to its own environment. In this talk, I'm going to sort of just outline some of the philosophy of our lab. We are interested in understanding the rules and principles behind regulated organization of the membrane. What are the principles of physics, chemistry, and genetics that we can apply? And how does the cell actually use this in order to perform certain tasks? What we know of the cell membrane is that it is a fluid mosaic, right? And this picture comes from Singer and Nicholson, more than 50, well, very close on to 50 years from their famous synthesis in this article in Science that they published about the fluid mosaic model, where they synthesized a whole bunch of information from the field, which told them that the proteins in a cell membrane were embedded in a sea of lipids, which had a bilayer arrangement of lipids. And at that time, this was a very, very important synthesis. And the Singer-Nicholson model, I must say, has dominated our understanding of the cell membrane to date. It began sort of, the Singer-Nicholson, as I said, was a synthesis of multiple observations, a synthesis of almost 100 years of experimentation on the understanding of membranes. And it was a biophysical perspective. The idea that some of these biophysical properties of the membrane could indeed have functional consequences was exemplified when Kai Siemens and Van Meer showed that membranes in general of a cell could in principle have something that they termed phase segregation. And that phase segregation, they felt was a consequence of the composition of the membrane, and therefore it would have arisen from the very fact that membranes that have multiple components can indeed phase segregate, and this is a well understood biophysical property of phase segregation, of thermodynamic phase segregation. And so the idea that you had these membrane domains, I mean, I will not use the term raft, but you have these membrane domains, that these domains could phase segregate was a property that they proposed was a consequence of the membrane composition. Mike Edidin, sort of much later, recognized that the membrane of a cell doesn't simply exhibit phase segregation, because otherwise, you'd have the same sorts of domains everywhere. But indeed, the membrane of a cell has multiple different types of domains. And so he said that the membrane of a cell has this patchwork quilt kind of a model. And he made a sort of a very prescient comment about 20 years ago, where he said that we are now awaiting a new model that integrates numerous features of a eukaryotic membrane, which have emerged since these were first characterized. But this reflected, even at this time, 20 years ago, the fact that the membrane produced these different wonderful patchwork quilts, in some kind of an equilibrium picture. And I should add that the equilibrium picture states the following, that if you have a membrane and if it has a particular composition, that composition somehow will phase segregate, and that those phase segregated patches then give rise to some sort of function. But I think people forget the fact that that for phase segregation, you need to have at least two parameters fixed. You need to have both temperature and composition carefully controlled, or tension, or third parameter could be tension, carefully controlled in order to get this phase segregated patch. And therefore, one needs to do one needs to magically position the membrane into a particular state so that it exhibits this phase segregation. And this was the sort of the state of thinking about membranes, around 15, 20 years ago. And we, at this time, were looking at the organization of a lipid-anchored molecule called the GPI-anchored molecule, and asking how does this molecule, distribute in the membrane? Because if you took membranes from living cells, GPI-anchored proteins, happily phase segregated when these membranes were sort of cooled below their physiological temperature. So what we did was we put binoculars onto GPI-anchored proteins. Rather, we put fluorophores onto GPI-anchored proteins and asked the GPI-anchored proteins to tell us about themselves, tell us about what's happening to them in the membrane of living cells. And we used what I've been claiming that I'll tell you about until now. We used HOMOFRET as a way of determining the local organization of the GPI-anchored protein with respect to its neighbors. Because the argument about producing a raft is that molecules, if they are in a raft, will concentrate and produce, and actually they will concentrate and jump into phase segregated patches, and therefore, the proximity between these molecules should decrease. And we argue that if you had sufficient molecules in the membrane, if they're concentrated, we should be able to pick up that concentration by measuring the proximity between molecules that were fluorescent. I hope that argument is clear. If it's not, please ask questions. Okay, so to measure this proximity, we, as I said, used this FRET technique called homofret, where you excite fluorophores with polarized light, and then if you have a fluorophore that is capable of its same kind, which is capable of accepting the energy in the excited state of the photoselected donor, then that energy of the excited state will transfer to this neighbor, and that neighbor will now emit fluorescence in a orientation that is very different from what its initially excited donor was carrying. So that orientation gets lost. And the way you measure this orientation loss is by measuring the fluorescence anisotropy, or the fluorescence polarization of the emission of the fluorophore. And how you do that is in a microscope is again very, very straightforward. One can do that by exciting a fluorophore by simply putting a polarizer in the excitation path of a microscope, and then sending the polarized light into a sample, for example, here. And in the polarized light, you then collect the emission from the sample into two orientations, both perpendicular and parallel. Sorry, the tags are missing here, but this is the top one is perpendicular, the bottom one is parallel. And then by simply doing this arithmetic of I parallel minus I perpendicular upon I parallel plus 2I perpendicular, you can get the anisotropy map of the fluorophores in a little patch of membrane. Now, if you remember, if molecules come close together, one would expect the polarization to be low. That is, if you have more molecules in a given patch, you'd expect a low fluorescence anisotropy. And if you have less molecules in a given patch, you'd expect high fluorescence anisotropy because the GFP molecule itself rotates very slowly. This kind of fluorescence imaging, polarization imaging, can be implemented in a wide range of microscopes, in TIRF, in total internal reflection microscopy that you've heard about, in wide field microscopy, in confocal microscopy, in multiphoton microscopy, in line scanning, and many others. So it's very versatile. You can measure this kind of FRET in all these different sorts of microscopies. So what we did was, take this a little further, asked whether you see FRET between these fluorescently tagged GPI anchored proteins that were expressed in the surface of membranes of different of living cells. And so here is the fluorescence of a GPI anchored protein, of a GFP GPI in the surface of a living cell, and its corresponding FRET map. And what you see is these very cool blue pixels of very low fluorescent anisotropy. And you see some orange pixels as well of high fluorescence anisotropy. And so that immediately tells us that there is a big difference between the organization of these GPI anchored proteins in these little patches, which are deep blue, versus these patches which are orange. And the difference, because we're measuring anisotropy, must be the proximity between different GPI anchored proteins in the membrane. And so by doing experiments that allowed us to, for example, if you photo bleach or remove the fluorescence of the GPI anchored protein by titrated dilution, you can actually model that change in fluorescence anisotropy and determine what kind of structures contribute to the lowering of the polarization. And to our surprise, when we made these measurements now, more than 20 years ago, we found that there were very tiny little clusters of GPI anchored proteins that were contributing to this lowered anisotropy in these patches. And these GPI clusters were concentrated in these patches. It was not as if they were forming one big patch, but it was as if they were forming small little clusters which were concentrating in those little patches. And I must say, for your information, this square box is on the scale of about 3 microns. And so these little patches where GPI clusters are highly concentrated are on the optical scale, meaning on the 250 to 1 micron scale. So you have these domains. Subsequently, using other methods, people showed that using in fact, Thomas who's on the call here, using near-field scanning optical microscopy, showed that you have clusters of GPI anchored proteins also produced in the membrane of living cells. But again, he looked at them in fixed preparations. And then subsequently, Jennifer Lippin-Schwartz mapped out with PALM, that is super resolution technique, showing that clusters are formed. We then, using methods that I'm going to describe shortly, showed that the clusters of these GPI anchored proteins could not have arisen by simply by GPIs sort of partitioning into phase segregated patches of membranes. One, because the clusters themselves formed in a concentration independent manner. That is, if you had molecules partitioning into a pre-existing phase, the partitioning would reflect would be based on the fraction that was present in and outside. That would be proportional to the concentration, and that was obviously not happening. The clusters themselves turned out to be immobile, spatially heterogeneous, as you can see here, and exquisitely sensitive to the presence of cholesterol in the membrane. And they were continuously remodeling. Okay, so to study some of these things, we again deployed a dynamic fret method, where what we did was in a confocal, we set up imaging, fluorescence, and isotropy in a confocal, and bleached a patch of membrane of the GPI anchored fluorophore, and watched the fluorescence intensity decay and the fluorescence anisotropy of the GPI anchored protein increase. Now, these were small molecule fluorophore labeled GPI anchored proteins, and so we could bleach them and watch the fluorescence anisotropy of that fluorophore rise quite happily. And then if you switched the lasers off and waited, say, for about 100 seconds, we could again repeat this process of fluorescence recovery, followed by the recovery of the bleaching of the fluorophore, followed by the loss of fluorescence depolarization because the fluorophore bleaches. So remember, if the fluorophore bleaches, the clusters themselves would start producing monomers instead of having a few clusters, and therefore the anisotropy of the system would rise. Right? So this rise in anisotropy is reflective of the fact that the molecules are pre-clustered, and when it bleaches, the clusters are now bleached away. But if you wait for some time, the clusters reappear. So this, so then to understand this, we created a reaction diffusion type model, and then identified that in the membrane of the cell, there were many different types of patterns we could see. And if you just took the patterns where you made clusters upon reappear upon waiting a period of time, we could then fit this reaction diffusion model and determine the fragmentation and formation rates of these clusters in the membrane. And we could fit the data, as I show you here, provided we assumed a couple of things. One, that the clusters were immobile, and two, that the clusters themselves were very small, and when they fragmented, they went from either trimers or dimers to monomers. And therefore, and what we got was really excellent fits across a whole range of fluorescence intensities in the membrane. And then if we plotted this whole business across a temperature scale, a temperature gradient going from very low temperature, so, well, I should not be that low, but 15 degrees to 37 degrees, we saw this very extraordinary plot. If we plotted simply the rate of fragmentation versus rate of formation, the ratio of those two rates gave us this sort of incredibly odd looking plot. I mean, the ratio of those two rates plotted against temperature should have given us an Arrhenius plot if the system was behaving in a reasonable, well-mixed manner, which is what you'd expect from these equilibrium pictures of the membrane. But the system, of course, what we saw was this, pattern which was, completely oblivious to temperature from 37 degrees to 24 degrees. And then sort of a catastrophic loss in that fragmentation formation rates below 24 degrees. So this told us that the cell was actually doing something which was temperature sensitive to build this thing. And at this time, we realized that motor proteins actually exhibit such a function. And motor proteins, of course, work on cytoskeleton. And so we made a leap of faith and said, what happens if you now take the membrane of a cell and disengage it from the cytoskeleton? So membrane of a cell, if it's disengaged from the cytoskeleton, looks like this. It forms these blebs. And these blebs can also be tracked by our method. By our method of watching fret. So if you take a bleb membrane, a membrane that's blebbing, and watch the anisotropy of the fluorophore in the membrane, what you see as soon as the membrane blebs, that membrane now becomes red. And when the membrane is being retracted back, it now begins to become blue again because it is building clusters. So this told us that, and a bleb, at this point, we knew, and many others knew, that the bleb is actually a piece of membrane that is detached from the actin cortex. And so we could put, I guess, more than two, but 2 plus 2 and 2 and many, many 2s together, and come up with the following, at least coarse description of what was going on. One, it was clear that the clustering must be actively maintained, because that kind of a formation fragmentation plot could not appear from something that was formed by a well-mixed solution. Then two, it must be dependent on an active actin cytoskeleton, because it was not present on blebs. And in addition, this allowed us to do experiments where we perturbed the actin cytoskeleton using inhibitors of the actin cytoskeleton, and these clusters fell apart. Okay. It was also immobile, concentration independent, and sensitive to cholesterol. So keep all these factors in mind. We need to now understand how these structures are formed and build some kind of an explanation for it. Of course, the first thing we realized is that the membrane of a living cell is closely juxtaposed to this actin cortex. And this actin cortex is something that imprints the membrane. It provides the membrane a pattern on which things can move about. And when things move about when membrane molecules move about in this sort of highly fenestrated imprint, they experience Hop diffusion, which has been well described by many of our colleagues, especially Aki Kusumi and his group. But this is a passive imprint of a actin cortex. There's no way that this cortex itself, in this form of interaction, could give rise to clusters. So it was at this time that Madan and Kripa, they began looking at this data and said, look, what must be going on is that the molecules in the inner leaflet of the membrane must be engaging with very dynamic elements of this actin cortex, and that those dynamic elements of this actin cortex are indeed going to give rise to stresses on the membrane, which will drive molecules into form these clusters actively. And for that, all you need are two ingredients. So let me tell you what the two ingredients you need. You need short dynamic filaments in the background of a static mesh. And you need myosin motor activity, which will, in principle, do all this. It will align and advect filaments. It will create, of course, contractile stresses. And those, along with the treadmilling of the actin filament, will give rise to a dynamic aster-like configuration of these filaments. And of course, we were game for any explanation, because what we were seeing was so puzzling that it wasn't really clear to us how we could understand these structures. So, and I think it's always good to take people to think, about things, and without being fettered by experiments or fact, or who actually allow leaps of fantasy to come into providing an understanding. It's actually a good thing to engage with such people, because you get pictures from a very different part of your brain begin to congeal. And that's sort of what happened. And what the picture that they produced for us was the following: that in fact, these actin filaments with myosin were producing these massive clustering drives, which were driving clusters of molecules in the membrane. And we came up with some very good validations of this, and I'm going to take you through some of that. But we first needed to find these small filaments. And indeed, using some really very clever FCS experiments. Well, here I'm showing some experiments from SIM that Thomas has done, and experiments of FCS, I think, that he's done, but others have done as well, where they found that if you took fluorophores that could bind actin filaments, what we saw were filaments that had diffusion time scales on the order of filaments that would be about 100 to 150 nanometers in length. And this indeed gave us the courage, if you will, to go and ask even further questions. So here is some FCS that we used to determine the filament lengths using some back of the envelope calculations of diffusion mobilities followed by structure. So what we did was we decided we would take this theory at face value, that you had filaments of actin, motor proteins, and if those things came together and bound ingredients in the membrane that were capable of binding actin, then sort of magically clusters would emerge, and these clusters would effectively be the clusters that would be causing the clusters of the GPI anchored proteins. Of course, at this point, you must already have guessed that I've been hiding the fact from you that the GPI anchored proteins happen to be on the outer leaflet clustered, whereas the clustering phenomena that we're talking about is the inner leaflet. And I must say, when we published these early papers, I was surprised that our reviewers failed to pick this up. I mean, they were very generous otherwise, that we were talking about a clustering phenomena on the opposite side of the membrane, whereas the phenomena that we were observing was on the other side. Okay, it's a small distance, it's only 4 nanometers away, but still, it's in an independent leaflet of the membrane. Okay, but we built our system to ask whether you could cluster anything on the same leaflet of the membrane. And here Darius, working with Kabir and Abrar, reconstituted a contractile actomyosin machinery on the surface of a bilayer where they had actin elements that could be recruited. And what they saw were these beautiful patterns. They saw these patterns of filament bundles, linked apolar asters, and these very exciting polar asters, which we thought were the elements that were contributing to our clustering phenomena. And in fact, that's what it looks like here. These polar asters, form, here's myosin clustering, here's the actin clustering, and here is the membrane element that's binding the actin that's also getting clustered. And those get clustered in a manner that's hugely, overdrive compared to what you'd expect from simply a static myosin actin cluster. In fact, when the system sort of becomes static, these clustered lipids that are being driven into these patches now begin to dissipate away. And they will then achieve sort of the equilibrium distribution, which is simply that related to the concentration of the actin in those locations. But what you see in these lipid and actin-binding lipid membranes are a huge overconcentration of these lipids, which is a consequence of the energy that's being consumed in the system to drive molecules together. I mean, this really was, to me, the first proof that we were experimentally able to validate some of these theoretical arguments. Okay, so if this is happening, we argue that membrane proteins, which have actin-binding domains in their cytoplasmic leaflets in a living cell, should also exhibit clusters that resemble our GPI active proteins. I mean, this is what we argued. And indeed, that's what we saw. And if you took away the actin-binding domain and replaced it with a mutant that will fail to bind actin, now these transmembrane proteins which fail to bind actin were simply wandering about footloose and fancy-free with no clusters in them. That this was happening in the membrane of all living cells with endogenous molecules came from work from both Parijat and Ruma. I'm going to show you some work from Parijat because she took this and created a surface map of where these clusters are formed. And so what she found is that using some really nice experiments that were done by Parijat based on work from Maria Parau's lab in Barcelona, where you could use cartography maps, where you take identification of single molecule tracks at high density, and then sort of correlate them in time or scrunch them up in time. And what you see are visitation maps of these sort of single molecules. And they form these really fantastic patterns on the surface of cells. So this is CD44 telling us where it visits and likes to hang out at the surface of a cell. And it turns out that it visits and likes to hang out at the surface of the cell in regions where it itself forms these highly tightly nano-clustered patches in the membrane. And it so turns out that this is very important for the function of CD44, ability to both present receptors for signaling as well as for phagocytosis. Okay, so what I've told you so far is that we have the actin cytoskeleton in sort of a way that we understand, at least by way of explanation, are driven by actomyosin machineries to form these clusters in the membrane. Equally, GPI anchored proteins must be doing the same thing in the inner leaflet because their properties look very similar. So to ask how do GPI anchored proteins cluster in the inner leaflet, we sort of did a bit of chemical biology with Ram Vishwakarma and found that you could take GPI anchored proteins with different, or GPI anchors, if you will, with different anchor lengths, from long to short to saturated to unsaturated, to just lipid with long saturated tails. And we found that it was only those that had saturated tails, or whether it was this type or this type, they formed clusters in the membrane. And they formed clusters, of course, by virtue of interacting with something in the inner leaflet. And what was interacting in the inner leaflet were, again, long acyl chain containing phospholipids, like phosphatidylserine, which were forming these links across the bilayer. And now using atomistic simulations, again, Madan's group and Anirban Poli working in his group showed that if you simulated these systems, we could in fact find that even in homogeneous mixed membranes, if you begin to trap these lipids in one region, somehow, maybe by using this actomyosin machinery, one will then be able to trap lipids in the outer leaflet, even under conditions where these bilayers are in a homogeneous mixed phase. So this is complete, well, I mean, I'm going to get back to Singer-Nicholson in a short while. So this is basically what they were proposing, that the membrane itself was being trapped locally. Lipids of a specific type were being trapped locally, and they were able to now connect across the bilayer and link up with similar sorts of lipids at the outer leaflet and create these little patches. And we believe that those are the patches that the GPI anchor proteins are capable of forming. And because we were able to do all atom simulations, the lipid structure in those little nanodomains was also so-called visible to us. And they reflected little local liquid ordered patches in the membrane. So what we are saying is that in the cell membrane, wherever the GPI anchor protein patches are present, for example, these blue patches, those regions are likely to be more ordered than others. And indeed, we now brought back further experiments, microscopy experiments, looking at how a fluorescent probe could tell us about these ordered patches in the membrane. And here we go back to Katharina Gauss's work. And Katharina Gauss, a colleague of ours who unfortunately and very sadly passed away earlier this year. She was an extraordinary biophysicist, a dear friend as well, but she unfortunately succumbed to illness in Australia. And I think many of us miss her and her really deep insights into using fluorescence methods, biophysical methods, and imaging for measurements inside cells. I mean, it's all the kinds of things that we do together. So Katharina Gauss also established this method of taking a solvatochromic probe, where you can probe the wavelength of the fluorescence emission from this molecule, and to determine whether in that local region where it is fluorescing, whether the membrane is either disordered or ordered. If it was ordered, the fluorescent wavelength emission would be more blue shifted. If it were disordered, the fluorescent wavelength of the fluorophore would be more red shifted. So using such a property of the fluorophore, we determined what was the nature of the domains where the GPI anchor proteins were locally clustered into these nanocluster rich regions. And so we found that the GPI anchored cluster rich regions were often associated with regions where this Laudan fluorophore exhibited highly ordered membrane characteristics. That is, its fluorescence was blue shifted. And there is sort of quantitative data that actually confirms such an observation. Okay, so what I've told you then is that the membrane is sort of intrinsically heterogeneous with these clusters of different types of molecules associating with this dynamic cytoskeleton. So for example, any transmembrane protein with the potential to bind actin, to have an actin binding domain recruited to it, is going to form these sorts of nanoclusters driven by actomyosin mechanisms. And any lipid anchor, any self-respecting lipid anchor, which had saturated acyl chains, is going to link up to this machinery that's available to it at the inner leaflet, which is building these clusters in the membrane. And this is going to contribute, if you will, to, just a moment, this is going to contribute to the production of local liquid ordered patches in the membrane in real time. So now we're coming back to thinking about the membrane. And so we can think about the membrane, therefore, as consisting of multiple types of species of molecules that would have nothing to do with this actin, which would not interact with dynamic actin. Those species of molecules, we would call them inert, and they probably represent the bulk of what Singer and Nicholson was talking about, proteins sort of floating about in these well-equilibrated lipid bilayers. Then you have passive molecules like our GPI anchor proteins that link up to this dynamic machinery at the inner leaflet, which ends up patterning the membrane both chemically and physically into patches which are rich in clusters of one kind or the other. And then finally, you have drivers of such a machinery. patches of membrane of receptors that would actually drive the production of such systems. And that's, you know, story for another day, but that's what we think are these integrin-like molecules. In fact, any signaling receptor will do that. So the membrane itself, we believe, is patterned in this way. We now know this, and I will again not take you to some of the data today, but tell you that we understand fully well how these patches arise by the generation of these actomyosin machineries triggered by the activity of membrane receptors that are activated by ligand. So when membrane receptors get activated by ligand, they activate Rho GTPases. These Rho GTPases, in turn, they produce filaments of actin, activate myosin motors, and create patches or clusters of actomyosin, which drive the assembly of these domains in the membrane. In turn, experiments, again, too long-winded to go into here. In turn, these membrane domains provide functionality to the membrane to influence the very receptors that gave rise to their being. They also provide functionality to the membrane to do other things. So for example, since these domains contain patches of liquid-ordered patches, they can support machinery inside the cell that prefers to hang out in liquid-ordered patches, because the liquid-ordered patches are now built locally. And then there, some new biochemistry happens. For example, the entire spreading reaction of a cell requires the activation of RAC. And RAC is activated when it arrives in these liquid-ordered patches. So, so now we have a way by which the cell can orchestrate the local activation of RAC by generating these liquid-ordered patches. So, and I think these are the sorts of things that I believe are giving rise to this incredible, you know, active bioarchitecture, if you will, that allows the cell membrane to act as this sensory apparatus, which all these things, interesting things, including the engulfment of these bacteria. So I hope I've convinced you that what we are seeing is a very dynamic system. We are seeing a membrane which is capable of sort of being self-organized by these you know, conductors, if you will, of an orchestra, the membrane receptors, which contribute to the self-organization, and that self-organization itself then has consequences for cell functions. For example, in cell migration, protein organization, and several others. Okay, so so basically that's sort of the story that we have come up with by wanting to pursue the structure of these GPI anchored proteins at the nanoscale in the membrane. Using the tools of microscopy and imaging, we've actually unearthed a completely, you know, new and different and distinct, exciting world of organization that is templated by active machinery, which we think is going to occupy us, at least for as long as I'm running a lab. But I think it'll probably last longer than that, because we're just beginning to understand the implications of what this means to receptor function and cell biology. So let me end by, again, sort of putting up the host of players who've been involved. I actually did indicate them at the beginning and did show you pictures of people as we went along. And I'd like to thank you for your attention. I know we've run a couple of minutes over time, but if you would like to have questions, I'm happy to stay and answer them. Thank you. I want to keep myself focused on this slide.
Thanks, Jitu. Does anyone have any questions?